Okay, so I think it's about time to begin. I want to welcome all of you to the final section of Bio 1B, and also express that you guys are real troopers to be here today. I know you've got your midterm for the ecology section tonight, but after that we'll be 100% into this final section on plant and fungal diversity. My name is Bruce Baldwin. I'll be the instructor for this part of the lecture part of the course the last few weeks here, and this is a part of the course that is going to deal with organisms that probably many of you have not studied previously. And I'd just like to get a brief show of hands of how many of you might have had previous biology course, even in high school, that covered plant diversity or botany in particular. Well, so quite a few of you. That's great. I know that it's often a hard sell to get people that have more of an animal or human focus to appreciate why this information is really critical for aspiring physicians, for people that are working in biology in general. But it's really important information that gets neglected too much, because when it comes right down to it, the plants and the fungi are really the foundations of our ecosystem. And without these organisms, we couldn't survive. There wouldn't be any animals. And so this is the group of organisms that includes the primary producers, the organisms that are actually capturing energy from the physical world and making it available to the living world through photosynthesis largely, and also the organisms that are recycling the nutrients that have been used by living organisms in the past. And without those organisms, those decomposers or recyclers, life on Earth would definitely come to an end. So these are really foundational organisms, and also more specifically, they really affect our lives in major ways as far as, for example, medicines. In the past, the botanists of the past, if you go back to the 1800s, for example, were mostly MDs, not PhDs, and to some extent that's even true today. If you take the medical ethnobotany course here in IB, which is a really great course, IB 117, taught by Tom Carlson, you'll be instructed by an MD who's actually a pediatrician at Kaiser in Oakland. And he follows in that old venerable tradition of physicians being botanists, because most of our medicines come from plants and fungi. If not directly, then our modern pharmaceuticals were inspired by secondary compounds produced by botanical organisms. So there's a lot of medical application of botany, as well as to diet. And we just look at one another. Me standing up here with my arms dangling at my sides, our hands. I mean, we're basically secondarily terrestrial creatures that most of our ancestral history as primates was spent in the trees, using our hands to grasp onto the limbs of plants. So we're all botanists one way or another, and ancestrally we're all tree huggers, literally. And that's really part of our, inextricably part of our biology. So as far as the vital stats of the course go, here's posted up here in my office hours, which are going to be directly after class, from 9 to 10 every day in the Bio 1B room right next to Mike Moser's office. And if you can't make the office hours because of a conflict, then please contact me by email, and I can set up a time to meet with you another time. And I'm happy to do so. My office is down in the university in Jepson Herbaria. So I'm a professor in IB, but I'm also a curator of one of the botanical museums on campus, which is the Jepson Herbarium, which focuses on the native plant life of California. And you may not be aware of it, but California is actually the biodiversity hotspot for plant life in North America, north of the tropics. And it's one of the biodiversity hotspots of the world. And so there's a lot of interest in the flora of California on the campus at large, and that's really the center of focus of the museum. All right, so my lectures are going to be posted on BSpace within 24 hours of the lecture, and you'll be able to find the PowerPoints there with the associated notes. So that'll be a good review, and of course the lectures are all webcast and available on demand. The readings are already posted for the syllabus, so just go to the icon for the plant and fungal diversity part of the course, and you'll see the whole list of readings for the course in the latest edition of Campbell. And I really strongly encourage you to do the readings, which helps to really reinforce the lectures and put them in a more detailed context, which can really be helpful for learning the material, because we do have to go fairly quickly over things in class, but the readings will really help to flesh out a lot of additional context that make the material more interesting and accessible. But the exam material is going to be strictly from the lecture. So I won't be testing you on subjects that are only addressed in the readings, which sometimes digress a bit from the lecture material. But I still think it's really valuable for you to read those assigned readings. So are there any questions at this point? Okay. So today we're going to start out with sort of a general introduction to the first question of what are plants. I'm not trying to insult your intelligence here. We may all think we know what plants are. We know one when we see one. But it's actually not such a simple question to answer. And the reasons for that are mostly historical. But after that, we're going to get into the first major group, the group that's most closely related to us, the animals, namely the fungi, as far as the botanical organisms go. And these are some of the strangest and most bizarre of the botanical organisms. We'll be first focusing on some of their general characteristics, and then next time we'll go into a lot more detail about their biology, their diversity, and their utility. Okay, so up until about the late 1960s, this was sort of the view of life on Earth, that we had the animals and we had the plants. And that's how most people classified life in general. And that goes all the way back to the ancient Greeks. I mean, this is basically Aristotle's view. And it carried forward to the time of Linnaeus, who was the father of modern classification of organisms or taxonomy. And really up until, like I say, the 1960s, this was pretty much the way people looked at it. Namely, that the animals are the animated organisms, the ones that have soul, that have perceptive sensory capabilities, that feel pleasure, pain, have imagination and desire, that can freely move about of their own will. That was kind of the sense that we have these organisms. And then we have basically everything else. And so the plants were sort of the residuum of the things that weren't animals. And basically plants were perceived as organisms that are alive. They're not minerals, which was the other way things were classified. They're living things that have the reproductive and nutritional capabilities, but they didn't have any sensory perceptive capabilities, and they definitely couldn't move about freely. They were literally planted in place. So that's the way that we looked at life on Earth in terms of this major dichotomy. And the science that was focused on animals, and this is still true today, is called zoology. We have a museum in this building called the Museum of Vertebrate Zoology, and that's a very time-honored field. Integrated biology was actually the Department of Zoology before it was reorganized back in the late 1980s. And then the plants, so the study of plants, all these basically non-animals, it's called botany. And botany is still a widely used term to basically represent the study of all these organisms. Oftentimes bacteriology is separated as a distinct field, but in the traditional way of circumscribing the field of botany, it actually included bacteriology as well. And if you look at the outside of the building, you'll see zoology and botany and bacteriology emblazoned up on the facade of the building. It goes back to the 1930s. So in the late 1960s, a view that had been emerging for really over a century became really widely accepted. That is, that life could be categorized into 5 major kingdoms. And those kingdoms were basically the animals, more or less as they had been classified before. And then these organisms that had been within the sphere of botany got split out into 4 major kingdoms. They were still considered the study subjects of botanists, but they were treated in 4 kingdoms. So the plants basically became restricted to the multicellular, mostly macroscopic or larger photosynthetic organisms. These organisms that are capable of fixing carbon. And so those are the, with the aid of light energy. And the fungi are basically the macroscopic multicellular botanical organisms that are not photosynthetic. And then we had the unicellular life, the microscopic life that was broken up into 2 major groups. The protista, which represented, like the animals, fungi, and plants, the eukaryotes that are unicellular for the most part. And then we have the prokaryotic unicellular life, which was all treated within one kingdom, the Monera. This is the way it was taught to me when I was in school, in college, and that persisted up until just very recently. And the reason we changed from this, in large part, was the rise of molecular biology, the ability to sequence DNA readily using PCR, and I know the details aren't important, but basically molecular biology became much more robust when we were able to amplify DNA readily using polymerase chain reaction and then sequence that DNA readily. And when this was done, okay, so let's, before going on to that in great detail, let's just review briefly what prokaryotes and eukaryotes are. So eukaryotes, for these 5 kingdoms, eukaryote literally means true kernel. The organisms that have these true membrane-bound organelles, the so-called kernels inside the cells, these membrane-bound units like mitochondria, where cellular respiration mostly occurs, and also in case of photosynthetic organisms, plastids, where photosynthesis occurs, as well as some other organelles. Eukaryotes possess those membrane-bound units and represent the protistans, the animals, the plants, and the fungi, whereas prokaryotes lack the nucleus and organelles. But the emerging view that came from the ability to sequence DNA readily, and especially from the open environment, environmental PCR, where we could take samples of soil or samples of seawater or fresh water and look to see what was there, even if we couldn't visualize it even microscopically, it became clear that there was much more life out there than had been previously described. And when it comes to the prokaryotes, this was a really dramatic revelation because it became clear that these monerans, these so-called monerans, which also were called bacteria, you know them as bacteria, were vastly more diverse than anyone thought previously. The only way that bacteria were ever previously recognized and described as classified organisms in our taxonomy was if they could be cultured on an artificial medium, for example, in a petri dish. That was how we were able to recognize bacteria. But it turned out that the vast majority of prokaryotes cannot be cultured. But we know that they're out there because when you sequence DNA from seawater, from soil, from all these environmental samples, you find the sequences of organisms of vast diversity. In just one handful of soil, you can find more prokaryotic diversity based on DNA sequences than you find in the entire classification system of all prokaryotes. So it's just off the scale in terms of the amount of diversity that's out there. And it became clear that actually this group that we thought of as more or less a singular group, the prokaryotes, represented 2 major domains of life, major clades, that are not even each other's closest relatives. And these are the bacteria and the archaea. And this group archaea is the one that we didn't really previously appreciate to the extent that we do now. And through phylogenetic analyses, it became clear that the archaea, also looking at their ultrastructure and looking at their biochemistry, it became clear that they share a lot of features with the eukaryotes that are not shared by the bacteria. But if we look at those membrane-bound organelles, we find characteristics, as we'll talk about a lot more in the next couple of lectures, that show very great similarities to bacteria. So we eukaryotes have within us features of both the archaea and the bacteria, and there's a really great reason for that. We'll talk about it soon. But in any case, there are these 2 major clades, and with regard to if we set aside our organelles and look at the rest of our cellular physiology structure, we're very much like the archaea, and that's because we do share a closer common ancestry with regard to those components of our cells than we do with the bacteria. And if we look back at the history of the fossil record, the very first evidence of life are really these prokaryotes. They go back 3.5 billion years, shortly after the formation of Earth. We have stromatolites, which are basically accretions of prokaryotes mixed with sand and inorganic matter that agglomerated onto the sticky biofilms produced by these early prokaryotes that show us that life on Earth was really quite ancient, and that these prokaryotes really precede the evolution of eukaryotes by almost 2 billion years. So a long time. All right, so these archaea, this group that's in many ways sister to us, the eukaryotes, includes some of the most fascinating organisms on Earth. And they grow in environments that other self-respecting eukaryotes or other self-respecting life forms, and all self-respecting eukaryotes would be killed by. Basically, these extremophiles, which basically means lovers, phile means lover, lover of extreme conditions, occur in environments that are really off the scale in terms of what most organisms can survive in. And these are broken into 3 major sort of functional groups. These are not natural clades for the most part, but just ways in which different extremophiles make their livings. Some extremophiles are bacteria, but the archaea are especially well represented among these organisms. Halophiles, halo is basically Greek for salt or saline conditions. So halophiles are organisms, these prokaryotes live in highly saline environments like the Great Salt Lake in Utah or the Dead Sea in the Middle East. So environments that are even saltier than ocean water, several times saltier in many cases. And not only do these organisms love salty environments, but many of them will die if they're in situations even as dilute as seawater. And some examples here that you might be familiar with if you've flown over San Francisco Bay and looked out, especially if you're flying into Oakland International Airport, you look down at the South Bay on the east shore, these commercial evaporating ponds that are set up along the edge of the bay here, evaporating seawater. That reddish coloration you might have noticed is actually the pigments of archaea. So infinite numbers of these organisms thriving in those salt ponds. And also another type of extremophile well represented in the archaea are these thermophiles, and these are named for growing in extremely hot water or hot environments that are liquid. And great examples of these are these archaea that grow down at these hydrothermal vents at the ocean floor, these little mini ecosystems that are completely supported by the chemicals being extruded from these black smokers, these hydrothermal vents, generally hydrogen sulfide, which some of these archaea use as an inorganic energy source. They oxidize the hydrogen sulfide, and the energy that's released then is used to fix carbon. So rather than photosynthesis, these guys are undergoing chemosynthesis and are producing the food that's necessary to support this whole ecosystem. These tube worms, these giant tube worms you might have seen in shows about this on PBS or something, they don't even have mouth parts. They feed by association with archaea that live inside of their tissues and are producing the food that they need to survive. So once these hydrothermal vents shut down, this whole ecosystem collapses. This is like the sun of this ecosystem. The sun to us, the plants capturing light energy that allows us all to live. These hydrothermal vents are basically the equivalent source of energy for those ecosystems. And here you can see a volcanic vent from Japan, where you can see this mat of archaea living in a volcanic vent of all places, incredibly toxic, sulfur-rich environment. But these guys are actually using those sulfur compounds to survive. So that's their energy source. And finally, there are methanogens. These are organisms that live in completely anaerobic conditions. Yeah. Yeah, there are a lot of organisms that can live in very cold environments. The thing we don't consider those extremophiles because the cold environments, one thing that these environments have in common is that these environments are so extreme that they will cause proteins and membranes to denature. And so cellular life is actually normally completely impossible in these kinds of environments. Whereas in cold environments, membranes and such can be disrupted definitely with freezing, but they're not as unstable as they are in these kinds of environments. Yeah, good question. Thanks for asking that. Methanogens are organisms that live in completely anaerobic conditions, oxygen-free environments in the guts of animals, and they produce methane in the course of digesting materials that are metabolizing materials within the guts of ruminants like cattle and other ungulates that allow those organisms to survive. So one of the things that I didn't even mention early on about botanical organisms, and especially microbes that have often been considered within the sphere of botany, is that we depend on them for our digestion. And with regard to methanogens, we also depend on them in processing of sewage. So in sewer plants, methanogens are really important for processing of sewage that we definitely need to deal with, and especially our highly compact societies. All right, so finally, one sort of an esoteric point, but it's kind of interesting to think about, is that these extremophiles have really allowed us to make the advances that we've made in molecular biology. So our knowledge that these organisms exist and are so diverse has come in some part from these organisms themselves, because these organisms, in particular these thermophiles, they grow in hot springs, like the Yellowstone hot springs, the geyser basins. These are the organisms that were the sources of our DNA polymerases that are stable at high temperatures. That's necessary for performing reactions like PCR, because we have to heat up the solution to denature the DNA so we can anneal the primers, et cetera, to allow this process of amplifying DNA to occur. And the only way that's possible is by using a polymerase that can withstand that kind of heating. So the proteins have to be stable, and we've extracted those proteins and then mass produced them from originally these thermophiles like Thermobacillus aquaticus. So there's some circularity here. The fact that we know so much about these organisms is in part because of them. All right, so let's move on to the bacteria then. The bacteria, we could spend an entire course on this group of organisms, incredibly diverse, a distinct group from the archaea, but similar in many ways. They're prokaryotes as well. And they include a number of well-diagnosed clades that have been recognized. And we're not going to get into those clades in detail, except for one. And that's the cyanobacteria, which were called the blue-green algae historically. And they were thought of as plants because they can undergo photosynthesis. So they can convert light energy to sugars. And so and in fact, as you'll see later, the cyanobacteria are actually the only organisms that have originated photosynthesis in the way that it's expressed in eukaryotes. All of our eukaryotic photosynthetic organisms, the plants, and some of these algal groups we'll talk about later, they owe their ability to photosynthesize to the cyanobacteria directly. We'll talk more about how that's possible shortly. Not today, but soon. So these are really the crucial organisms. Many of these organisms are crucial. The bacteria also include a lot of decomposers, organisms that recycle nutrients that otherwise would eventually all be tied up in compounds that we couldn't utilize. But they also are capturing light energy and converting it to sugars. And they're